In this video, we are going to specifically talk about the reactions that aldehydes and ketones undergo. We're going to start by discussing nucleophilic addition reaction. The carbonyl carbon that is present in these two functional groups is electrophilic. This is derived from the resonant effects and inductive effects that the carbonyl carbon has. First, when it comes to resonance, as you can see, the electrons that are present in the carbon-oxygen double bond can be placed onto the oxygen atom. That creates a negative charge on the oxygen, while the carbon can be positively charged. So there we can illustrate the electrophilicity that the carbon in the carbonyl has. The inductive effects that we observed is because of the polarity that we previously discussed when we introduced the functional group, establishing that the oxygen atom is more electronegative than the carbon atom. So if we put the partial charges for both atoms, as you can see, the oxygen is more electronegative, the carbon is more positive, and again, it's because of electronegativity value differences. That means that when it is attacked by nucleophiles, understand that the new sigma bond is going to be specifically attaching to the carbon atom, and I'm going to draw it into my carbonyl. When that happens, now we're going to be forming a new sigma bond, which I'm highlighted in green, between the nucleophile and the carbon in the carbonyl, and the electrons are then placed on the oxygen atom, and as you can see, it becomes negatively charged. Overall, once these nucleophiles attack the carbon in the carbonyl, then that center becomes an sp3 hybridized center, and it is tetrahedral. Now, remember that both of these functional groups are going to have carbonyls. When it comes to aldehydes, they are generally more reactive towards nucleophiles than ketones. One of the reasons are steric effects. Remember that aldehydes are going to be less sterically hindered because along with the bonds that we have attached to the carbonyl, in aldehydes we have a hydrogen. Also, there are electronic effects. Aldehydes are going to have a large delta positive on the carbon of the carbonyl. Okay, so if we consider the structure of aldehydes and ketones, as you can see in the bottom left corner of the slide, a ketone is going to have two electron donating groups, because remember that the R groups that are present in the ketone are alkyl groups, and that stabilizes the partial charge that is found on the carbon in the carbonyl. When we're talking about an aldehyde, remember, we only are going to have one R group, if any. So in this example, we're considering that the R is going to be alkyl, and having that alkyl group in the aldehyde, and again, this is not taking into account formaldehyde, then we're going to have only one electron donating group that is going to that's going to stabilize the partial charge that it is in the carbonyl. Some nucleophiles that are going to be reacting with our functional groups are going to require acidic conditions. Others are going to require basic conditions. So, one example of a nucleophile that is going to require basic conditions is a Grignard. When it comes to the Grignard, the alkyl group is going to have a carbon anion, and it's going to add to the carbon atom in the carbonyl. Okay, the result of this reaction, as you can see, is going to be an alkylated alcohol. So in general, understand that a strong nucleophile means that the reaction is going to be under basic conditions. So examples of those strong nucleophiles include Grignard reagents and hydrides, to just mention a few. Under basic conditions, all our nucleophiles are going to react with carbonyls by the same general mechanism. So what happens is that our nucleophile is going, and I'm just going to highlight the arrows as I mentioned them. In the first step of the reaction, our nucleophile is going to go and attack the carbon in the carbonyl. The electrons are going to go up to the oxygen. Once that occurs, in the second step of the reaction, the oxygen that is negatively charged is going to undergo a proton transfer. As you can see, it's been illustrated by water. That means that water is going to lose a proton. The electrons are going to go back to the oxygen, and that's how we are forming an alcohol. So understand that when the nucleophile attacks, it's going to be forming that negatively charged intermediate. That in order to make it more clear, this negatively charged intermediate is present in this molecule. And then we have to work it up under acidic conditions, which is what we observe with the proton transfer. Aldehydes and ketones both react with a variety of weaker nucleophiles also, but they do so under acidic conditions. And they have the same general mechanism. When this occurs, as you can see, the first step of the reaction, I'm just going to highlight the arrows, are that specifically the oxygen in the carbonyl is going to be protonated, and then the electrons are going to go to the other atom that contains the hydrogen, okay? Again, in that proton transfer, we are going to have a molecule that is going to allow this protonation to occur. Then, in the second step of the reaction, we're going to have a nucleophilic attack. So that protonated carbonyl is going to be attacked by the nucleophile at the carbon of the carbonyl. Then the electrons are going to go up to the oxygen. Again, during these conditions, we're going to have a similar product. We're going to have now an alcohol that is also going to be substituted by the introduction of that nucleophile. Now, understand that the difference between acidic conditions and basic conditions is that, as you saw previously, under basic conditions, our intermediate is going to be negatively charged. And when we are under acidic conditions, then the intermediate is positively charged. As you can see, the molecule that I underlined in blue. And again, the difference between the two is also the strength of the nucleophile. So, understand that in order for these weak nucleophiles to attack our carbonyl, we need the acidic conditions for the protonation of the carbonyl, okay? The protonation actually helps the whole system by making our carbonyl a better electrophile. And as you can see in the following drawings, once we have the protonation occurring, and this is the intermediate that we first proposed, it has a resonance structure in which now you can observe that we form a carbocation specifically in the carbon atom in the carbonyl, and the oxygen is neutral. So as you can see, protonating that carbonyl makes it a much better electrophile, and since our nucleophile is going to be a weak nucleophile, that's why that is more advantageous. When a nucleophile attacks a carbonyl group, understand that the equilibrium is going to depend on the ability of the nucleophile to function as a leaving group. So let's take, for example, that we have the following molecule. Let's just assume that it is a ketone. And if we put it under acidic conditions, as you can see, we have an arrow that illustrates the equilibrium. Since the nucleophile in this system is going to be our chloride ion, okay, and is also a good leaving group, understand that the equilibrium in this case is going to favor the starting ketone. So when we consider these arrows, instead of being in equilibrium between reactant and product, is going to favor the ketone. But again, it comes from the idea that this chlorine that was added to the ketone is also a good leaving group. So when it comes to this, the ketone is the one that is going to be favored. So understand that in this chapter, we are going to cover the following nucleophiles and their reactions with ketones and aldehydes. So as you can see, in the first column, we have oxygen nucleophiles, followed by sulfur nucleophiles, then nitrogen nucleophiles. We're going to cover hydrogen nucleophiles, and lastly, we will be going over carbon nucleophiles. Let's start first by covering the oxygen nucleophiles. So understand that in this case, we are going to initiate by talking about with OH as a nucleophile. So the basic mechanism, then this is illustrating the base catalyzed hydration for an aldehyde. We are going to have hydroxide as a nucleophile. So what happens in this reaction is that in the first step of the reaction, we have a nucleophilic attack of the hydroxide ion onto the carbon in the carbonyl, illustrated by that first arrow. I'm just going to highlight it to make it more clear as I mention it. So the hydroxide ion is going to attack the carbon in the carbonyl. The electrons are going to go up to the oxygen. In the second step of the reaction, we're going to have a proton transfer in which the oxygen atom gets protonated, and as you can see here, it is base catalyzed because we are going to be regenerating specifically that OH ion, because, or that polyatomic ion, because we are going to be utilizing water for that protonation step. Okay. Understand that when we have oxygen nucleophiles under acidic conditions, specifically, the carbonyl is going to be protonated, and then water is going to be our nucleophile. So to describe that mechanism, in the first step of the reaction, we're going to have the protonation of the oxygen in the carbonyl by hydronium providing a proton. Then that means that now the aldehyde is going to be more electrophilic. So now our nucleophile, which is going to be water, is going to be able to attach to the carbon. In the carbonyl, the electrons are going to go up to the oxygen. In the next step of the reaction, understand that what's going to happen is that now water is going to act as a base, is going to remove a proton from the intermediate that formed when our nucleophile attacked. That's going to bring the hydrogen-oxygen bond, and as you can see, that intermediate that was deprotonated, which is called the oxonium intermediate, okay, is going to form this molecule that is called a hydrate. I haven't established the name of that compound, but if we follow through, as you can see, when we had either OH or when we had hydronium, we are going to specifically form two OH groups attached to the carbon atom that it was part of the carbonyl system. Another set of oxygen nucleophiles understand that has to do with alcohols. Alcohols can attack ketones and aldehydes. Now, understand that if we have two equivalents of our alcohol under acidic conditions, we are going to form an acetal, and one of the byproducts is going to be water. So, if we are under acidic conditions, again, that ketone or aldehyde is going to react with two alcohols to form that acetal. In this reaction, we have some commonly used acid catalysts to form the acetal, and they include para-toluene sulfonic acid, or TSOH, or sulfuric acid. Now, the mechanism for this reaction, as you can see, is as follows. This is an important reaction in biochemistry. The reason why I mention it is because this actually is what gives rise to many sugars or carbohydrate motifs that are present in or that are present in the human body. But understand that this is the way that the mechanism goes. Here we are going to be utilizing a general ketone in order to establish the reaction itself. So the first thing that happens, because we're under acidic conditions, is that we are going to have the carbonyl group, okay, going to be protonated to make it more electrophilic. Then our alcohol is going to attack the carbon in the carbonyl, and the electrons are going to go up to oxygen, okay? As you can see, that is going to make the oxonium intermediate. In the next step of the reaction, okay, we are going to have A minus, which in a way that's going to be the conjugate base that resulted from the first step of the reaction, is going to deprotonate that oxonium intermediate, and is going to give rise to a hemiacetal. So understand that the hemiacetal is an important intermediate. It's like the halfway point to getting to the acetal. Then, as you can see in the next step of the reaction, we're going to have a second proton transfer, but at this time what we're going to be protonating is the OH group that initially, if we keep track of the atoms, this oxygen, which became OH in the first step of the reaction, remains OH here, and in our hemiacetal as OH, is what gets protonated, because as you can see, once we protonate it, what's going to happen is that we are going to lose the leaving group, meaning that water is going to lose the system, and this is an intramolecular reaction in which the oxygen electrons are going to come down and are going to regenerate that C double bond O, and that's why the water gets stopped. After that step, then a brand new alcohol molecule attacks the carbon in the carbonyl. Once that happens, we are going to have the electrons go up to the oxygen, okay? And we're going to be generating another oxonium intermediate. And in the last step of the reaction, as you can see, the A minus, or the conjugate base of the system, is going to deprotonate That's oxonium ion that was formed after the formation of the hemiacetal in order to generate the acetal. Understand that the acetal formation is an equilibrating process. For most simple aldehydes, the acetal is favored at equilibrium. So as you can see here, we have an example of formaldehydes reacting with two equivalents of ethanol and is making the following acetal. Understand that for most ketones, the acetal is actually not favored at equilibrium. In this case, the reactants are the ones that are more favored during equilibrium. Now, understand that when it comes to this, we can actually, instead of utilizing two equivalents of the alcohol, then if we can get around that by utilizing in our reaction a diol, okay? And a diol just means a di alcohol. And as you can see here, this is an example of a diol. Understand that in this diol, what we're going to form is a cyclic acetal. And as you can see, that cyclic acetal undergoes the same mechanism that we explained previously, but now because the molecule has the two OH equivalents, then we're going to have a cyclic acetal. Make sure that you practice the mechanism for acetal formation when we have as our alcohol molecule a diol. Understand that acetal formation is actually a reversible process and it can be controlled by adding and removing water. To favor acetal formation, understand that water needs to be removed from the reaction. So there's actually instrumentation that is utilized in the laboratory in order to be removing the water and pushing the equilibrium of the reaction towards the formation of the acetal. Understand that to convert the acetal back to the ketone or the aldehyde, water is added to the acetal with the addition of an acid catalyst. In this way, acetals can be used as protecting groups for aldehydes and ketones. And this all has to do with the type of reactions and synthesis strategies that you're trying to utilize in order to make certain products. So let's consider the following reaction. Let's say that specifically you want to synthesize, okay, a particular molecule, okay? We need to convert, okay, an ester to a primary alcohol, okay? And typically in this process, understand that you could do that by using lithium aluminum hydride, okay? But the thing is that in this synthetic strategy, if you utilize those conditions, as you can see, our target product, you want that ketone to be unchanged. So the problem here is that lithium aluminum hydride does not selectively will reduce the ester to the alcohol. It will also reduce the ketone. So how do we go around that? Well, we can use specifically a protecting group, okay, for the ketone in order for when lithium aluminum hydride reacts with the molecule, The ketone, we're going to mask it. So it's kind of like we're going to put a hat on it in order for this reagent to not observe it during the reaction. So how do we go about this? Well, because we have a ketone, when it comes to making an efficient aldehyde, we can utilize a diol, okay, to make a cyclic acetal as the first step of the reaction. As you can see, we're going to use our diol under acidic conditions. We are going to remove water, and what happens is that now we are forming a cyclic acetal. Now, in the second step of the reaction, as you can see here that I highlighted in green, then we can add the lithium aluminum hydride, which is going to selectively reduce your ester to an alcohol. The third step of the reaction, that's just for the protonation of the alcohol. But understand that in this process, this step what it's going to do is remove the cyclic acetal also, because that's something that is very interesting about these protecting groups, that you place them in with acid, but you can also remove them with acid. So in that third step of the reaction, we will be removing the acetal, and that's how the ketone gets regenerated and our alcohol gets protonated. Now, understand that we mentioned during the process of discussing the mechanism that a hemiacetal is one of those intermediates that is made during the formation of the acetal. A hemiacetal, in general, even though we discussed that it is formed during the process of making an acetal, it's difficult to isolate. Okay, because in the whole process, the equilibrium is going to favor, okay, either the initial aldehyde or ketone or the acetal, depending on the conditions. The only ones, okay, that are going to be favored specifically is if you have a cyclic hemiacetal. And this is where I was mentioning that this reaction is actually very important in biochemistry, because cyclic hemiacetals are what we know as sugars or carbohydrates. They're an example of a cyclic hemiacetal.